Hello and welcome to another lecture to Bio 111. So in this lecture and in the next lecture, we will talk about cell division and what happens during cell division and the different types of cell divisions that can occur in cells. There are the somatic cell divisions that are just occurring to cells during regular growth of the body, where the body, the soma, the body, grows and divides cells, where each cell is identical to the next. That is accomplished by mitosis. And then there are the formation of the sex cells, egg and sperm, and that happens in meiosis. So today we'll talk about mitosis. The next time we'll talk about meiosis. So before we start talking about what happens in eukaryotes, I want to give a very brief overview of what happens in cell division in prokaryotes. So that is referred to as binary fission. So what you see on this slide is a bacterial cell, and bacterial cells have chromosomes that are circular. So the circular bacterial DNA is attached to the membrane, to the inner membrane, and it has a site where DNA replication can occur. So if you see that it always happens at the same place, the origin of replication. If you see here, there's now a replication process that has started. So DNA replication begins at a specific location, that's the origin of replication, and proceeds bidirectionally around the circle. So it can move both this way and it can move also in that direction. At some point, that circle is finished, is completely synthesized. The new synthesized DNA molecule is also attached to the inner membrane near the attachment site of the initial molecule. But they're still both in the same cell. So at that point, the cell has to divide so that it now has only one set of chromosome for each of the cells. And that can then occur after the cell has elongated a little bit. You get these formation of the two different cells, and in the end, a constriction that separates the two daughter cells. So in eukaryotes, cells follow something that's called a cell cycle, the cell cycle. So the cell cycle is divided into multiple different stages. There are really two main stages. There's the interphase, that's almost all of the cell cycle, and then there's the M phase. And the M phase stands for mitosis, essentially. So mitosis and cytokinesis. Cytokinesis is the cell division. Mitosis is the chromosomal replication, so the duplication in a very regulated fashion happens during mitosis, and then after that, the cells are divided in cytokinesis. Cyto stands for cell, kinesis stands for division, cell division. So we'll talk a lot about the M phase today, but I do want to spend just a little bit talking about the G phase or the interphase, the G phase, the S phase, and then the other G phase. So those together make up the interphase. When cells have divided, or you could say before they divide, they can enter a dormant phase. So that's the G0 phase. And that's in that phase, they just sit and they don't divide for a while. But if they are going to divide, they need to duplicate their entire genome. And that happens in interphase, in the S phase. So S stands for synthesis phase. And so after a gap, the synthesis occurs, and then there's another gap, that's the gap phase 2. And so during interphase, the cell waited for a while, could be a long time, but at some point when it's committed to going through cell division, it has to first copy the DNA so that it can divide that equally into the two daughter cells. And once it's committed and it's duplicated its DNA, and it's gone through gap phase 2, it will enter mitosis. And that is where the cell division and the sorting of the duplicated DNA then occurs. So when we look at chromosomes, we can actually see them in the microscope. depending on the size of the chromosomes, different organisms have different size chromosomes, we can create something that's called a karyogram. This is a karyogram of a human, and we can color them with a variety of techniques. But that's not really part of what we want to discuss, how we can color them. But we can make them more easily visible. So when you see them in the microscope, they're not colored, right? They don't have these colors. These are colors that, these are false colors, essentially. So they can come through the different ways of visualizing them. But they do help in order to pair them up. And the reason why we want to pair them up is that when you extract chromosomes, intact chromosomes from cells, and you put them on a microscope slide, of course, they don't look like this nicely sorted. They're a big jumble of chromosomes. And you see that they're sorted this way by the two identical chromosomes, well, identical in size and length and the arrangement of their genes along the chromosome. And that is what's called homologous chromosomes. So homologous chromosomes are the chromosomes that you get from your father and from your mother, one from each. And so they have the same genes on both sides, but there can be slightly different variations. And we'll talk about those variations later on when we talk about genetics. But for right now, let it suffice to say that we have a chromosome 1 from the mother and a chromosome 1 from father. They have the same length, they have the same general genes arranged from top to bottom along that chromosome. And you can see that in the karyogram, they are now sorted by homologous chromosomes. There's always two, with the possible exception of the sex chromosomes. So the sex chromosomes are the chromosomes that confer the sex of the organism, male or female. And in humans, males have an X and a Y chromosome, and females have two X chromosomes. And so the two X chromosomes are very similar. The X and Y chromosome are different from each other because the Y chromosome, which is not pictured here, is much smaller. So the way that a karyogram by definition, convention, is always sorted is the tallest chromosomes first, and then they get smaller and smaller. And at the very end, even if they're taller, you add the sex chromosomes. So these are all what's called autosomes. Autosomes are the non-sex chromosomes, and then at the end, the sex chromosome. So the karyogram shows what's called the karyotype. The karyotype is just basically how many chromosomes do you have in a cell of a given chromosome, and are they all there? Are they broken? Are they all intact? Are there additional ones? Are there any missing? That's what a karyogram shows you. And so what I write here on the right is that a karyogram pictures the karyotype. So that's the karyotype, nicely sorted in a karyogram. And they're used to determine certain chromosomal disorders. So you can, for example, pre-birth, take a sample of an embryo, and you can make a karyogram, and you can then see whether all the chromosomes are the way that they are normally found in a human, which looks like this for a female. Now, there should be 23 pairs in humans. Other organisms have different number of chromosomes. 22 autosomes in humans and 2 sex chromosomes. In humans, the sex chromosomes are typically XX and XY, and then there are a number of variations that you can find in karyotypes. And so the variations that you find, if the karyotype is different, usually leads to pretty stark phenotypic variations. In fact, in humans, having additional chromosomes or missing certain chromosomes is in many cases lethal. That's how important it is that the chromosomes are balanced, that there's one of each kind of chromosome. But there are some cases where certain chromosomes can be aneuploid. That means there's one missing, or additional, aneuploid, as opposed to euploid. We'll talk more about that later. But what I want to point out here is that among those potential variations in humans is the trisomy 21. So if there's a third chromosome here, that's known as Down syndrome. And then there are some variations that include a change in the number of sex chromosomes. So there can be a monosomy. Monosomy, if there's only one some really means body, so these are the chromosome bodies. Monosomy, just one of the X chromosomes. That's also referred to as Turner syndrome. And then if you have two X and a Y chromosome, that's called Klinefelter syndrome. So there are some differences that people with Turner syndrome or Klinefelter syndrome have to folks that have a double X or an X and a Y. Okay. So let's talk about some terminology that we need to understand in order to talk about meiosis and later on mitosis and later on meiosis. So I already said there are homologous chromosomes and one comes from each of the parents. So oftentimes in these diagrams you see them pictured in the same color but different shades. So they're both in blue, but this is dark blue, this is light blue. So let's assume that one of these homologous chromosomes in a particular cell came from the male parent, the father, and the female parent, the mother. They're both the same general chromosome. Let's say this is chromosome 1. Therefore, they are homologous chromosomes to each other. Homologous chromosomes have a middle point and they have arms. Sometimes the middle point is not really in the middle, but the middle point is called centromeres, regardless of whether it's exactly in the middle or not. It's a constriction where the DNA is very dense, very compact, and where the later on the fibers that actually bind to chromosomes and then yank them to the poles, you'll see that later on in some of the slides, where they attach. So it's sort of an attachment site. So that's the centromeres. Centromeres, these are homologous chromosomes. And then when they go through S phase, so when they go through the duplication phase, they duplicate the entire chromosome length. And then there are two of those, and they are also attached to each other at the centromere. So after S phase, these are still both chromosome 1. It's only one. This is one chromosome, number 1. This is one chromosome, number 1, from one parent and from the other parent. Therefore, they are still homologous chromosomes, but now they're duplicated homologous chromosomes. And when they are duplicated, they consist of two what's called sister chromatids. These two sister chromatids are exactly identical. There's no difference between sister chromatids after a chromosome has gone through S phase. And that makes a lot of sense because if you duplicate this from top to bottom, you're just making an exact copy. And then you're attaching it as it's being duplicated. It stays attached at the centromere. And so now we have a duplicated homologous pair of chromosomes. All right. So when a cell is going through cell division, it needs to duplicate its DNA. So now it's done that through S phase. And then it also needs to sort these chromosomes, one to the one daughter cell and the other one to the other daughter cell. And out of those, let's say 46 chromosomes in a cell, you can't just half them and say 23 go in one and the other 23 go the other. You have to make sure that there's a chromosome 1 that goes to one daughter cell and the other chromosome 1 goes to the other daughter cell. Otherwise, you end up with, maybe none of the chromosomes of a certain kind and 2 chromosomes of the other kind. So that is what mitosis does. So let's take a look at how that happens. So there are multiple steps that occur. And I'm just going to show you the overview and then we'll go through each of the steps individually. So these are pictures that are taken from cells that are divided and they are fluorescently labeled. So you have fluorescence that either highlights fibers and they are in green, or blue, that's the chromosomes. So the DNA is all in blue, and then these fibers, spindle fibers, we call them, they are in green. And so here's a cell in here. It's a little bit hard to see the exact outline, but you see that early on in what's called prophase, we'll get through the individual stages in a second, they condense and there's right now you can't really make out the different chromosomes, but here you can. So now you see that they become much more condensed, much more defined, if you will. And these spindle fibers that are all over the place here are now also becoming more defined. And they become even more defined in the phase that's called metaphase. And you see that all these blue chromosomes are aligning here in what's called the metaphase plate. And then later on in the last phase, well, in the next to last phase, really, they get pulled apart in what's called anaphase by these spindles to both of the poles, left and right. And then in telophase, you see that now we have a set of chromosomes here and a set of chromosomes here, separated by these green spindle fibers, and then they are starting to separate into two different cells. So I want to, for the overview, I want to give you a few things to remember, and that is that cells before and after mitosis are completely genetically identical. They have the same number of chromosomes. So if you start with if you start with 46 chromosomes, at the end you also have 46 chromosomes, and they have not exchanged any chromosome arms or DNA or any kind of information. They're completely identical. So that is important, because if a cell just grows like a skin cell, it has to be identical to the two parent cells. Now, how does it do that it has the same number of chromosomes as the parent cell that it had before? So let's go through the individual stages and look at this, how this happens. So the first stage is called mitosis. In mitosis is called prophase 1. And your book says chromosomes condense, centrosomes radiate, microtubules, and migrate to opposite poles. And so what I want to point out here in these drawings is that these are the centrosomes. So these are bodies that are forming from which spindle fibers start to emanate out, to sort of grow out. This is the entire cell. Here's your nucleus with a nuclear envelope, and inside you have the DNA. So it says here chromatin fibers. Those are your chromosomes. They're still attached to proteins, and so that's why it's called chromatin. But this is essentially what's going to become your DNA. And you already see that they draw it in the dark blue and the light blue, but it's completely jumbled. So this has to condense in order for the spindle fibers to attach to specific regions and then be able to sort them. In this jumble, it would be very difficult to get the right chromosome attachments and then pull them apart without them breaking. So what happens in prophase 1 is that these chromosomes condense, and then these centrosomes, they move to the poles and they make these spindle fibers. But the spindle fibers cannot yet attach to the chromosomes because there's still the nuclear envelope around it. So down here I have a little set of things that I would ask you to remember for each of the phases. You can read this. I basically already told you about this, but these are the things that you should remember for each of the phases. So this is prophase. In pro-metaphase, that's sort of between prophase and metaphase. Some folks in the past would call this late prophase or early metaphase, and sort of in between. So let's just call it pro-metaphase. You see that now the chromosomes are much more condensed here. The spindle fibers are now not really attached to every chromosome yet, but you see that they are forming, they're moving to the poles and they're forming these long fibers. And where they attach eventually, you see that here a spindle fiber has attached to the centromere. They're sort of attached to the centromere, but in this little graphic here, you can see that yes, they attach to the centromere eventually. This is a spindle fiber, one of those green fibers over here. But they don't directly attach to the DNA. They attach to something that is surrounding, like a sheath, the centromere, and that's called the kinetochore. So the kinetochore here in green, that's a protein complex to which the spindle fibers attach. And then they just sit there for now. So spindle fibers attach to chromosomes via the kinetochore complex to the centromere. Centromere being part of the DNA, kinetochore being a protein that is attached, wrapped around this centromere. And then the nuclear envelope finishes breaking down completely so that this attachment can fully happen everywhere. So then metaphase happens. And in metaphase, the chromosomes align in the middle of the cell, and that middle, if you will, of course this is a sphere, right? So exactly where's the middle? That's called the metaphase plate. You can picture that if you think of a globe, Earth, a sphere, where the equator is. So I say here, like the cell's equator. Of course, there's no real equator, but if there were one, that would be where the metaphase plate is and where these chromosomes align. And you see that they don't align with each other, right? There's one on each side, so they don't pair or anything. They're just sitting next to each other. And then you can see that the spindle fibers are starting to attach. They help in aligning these chromosomes nicely in the metaphase plate. And so then the next phase occurs, and the next phase is called anaphase. Of course, these phases are fluid, and the picture that you see here is sort of in the middle of anaphase already, where chromosomes have now started to be pulled to the side. These are called the poles, pole and another pole. So again, in that analogy of the Earth or a globe, we have the two poles, the north and the south poles. We have the equator here. And so the chromosomes are now being pulled by their centromere, by their kinetochore wrapped around the centromere to those poles. And at that point, they separate. The sister chromatids separate from each other. I say here, sister chromatids separate, and after separation, each original chromatid is now considered a chromosome. So at this point here, you already have 8 chromosomes in this cell. 1, 2, 3, 4, 5, 6, 7, 8. Whereas right before, we only had 4 chromosomes. And this is sort of a definition thing because the amount of DNA is the same, right? We had 2 chromatids here per chromosome, but 1, 2 chromatids, right, per chromosome, 1, 2 chromatids per chromosome. But we only had 4 chromosomes. That's a definition thing. So now we have 8 chromosomes, but we have no more chromatids because a chromatid is one of 2 arms of a chromosome. This doesn't have chromosome, shouldn't call them arms, halves of the chromosome. So the chromatids, they're no longer chromatids. There's still, the top and the bottom of the chromosome. It's just not duplicated anymore, and so therefore it's called a full-fledged chromosome with no chromatids. Okay, so then that happens in anaphase, sister chromatids separate. And then in telophase, the nuclear envelope reforms, the chromosomes condense, the mitotic spindle starts to disappear or sort of to go to the sides and then eventually disappear, but the cells have not yet divided. So in telophase, the cells do not divide. That happens in cytokinesis. What does happen is that the nuclei are now fully forming again with the nuclear envelope, and so therefore that has divided. But that is the end stage of mitosis. Telos in Greek meaning the goal, the final goal, and so that is sort of the final goal of mitosis, not yet of cytokinesis. That happens in the next stage, and then you see that cytokinesis in animals and in plants works slightly differently because plants have that cell wall, right? So they can't just constrict these 2 cells by a contractile ring as it happens in animals. You can see the membrane here forming, and that's in red. The DNA is in blue, the new nuclei here, and then we have cytoskeleton fibers in green. That contractile ring forms and sort of pinches off those 2 different cells. That cannot happen in cells because in plant cells because the plant cell walls are rigid, and so they put in something that's called a cell plate, and the membrane forms around the cell plate, and the new cell wall is produced as well. But you can nicely see, and you will see that in lab as well in microscopic slides, that you can see the chromosomes in the nucleus very nicely, and then around it you see the cytoplasm, cell wall, and so forth. Yeah, and so that is what happens in mitosis. So let's just sum that up. In eukaryotic cells, there's the cell cycle, and it's divided into 4 phases: the gap 1, the S phase, the synthesis phase, gap 2 and M phase. There's also the G0, that sort of the offshoot, off-ramp for parking, for long-term parking. And that is then in M phase, we have the mitosis and the cytokinesis. And then cell division in eukaryotes occurs by mitosis. There are these 5 steps that we just walked through: prophase, prometaphase, metaphase, anaphase, and telophase. Actual cell division is in after telophase, is in cytokinesis. During mitosis, sister chromatids are separated. And I bold that here because that is one of the main things that you need to take away from mitosis. What happens in mitosis? If you were asked that question, the answer is, during mitosis, sister chromatids are separated. Chromosomes are made up of 2 chromatids after they go through S phase, and the same numbered chromosomes that come from each parent are called homologous chromosomes. So we have pairs of chromosomes, say number 1 and number 2 and number 3, from each parent, and those are called the homologous chromosomes. So what happens in mitosis is that the sister chromatids of every chromosome get separated. And the reason why you can have the same number of chromosomes at the beginning in one cell as in the end in 2 cells is because the sister chromatids make the duplication, right? So you call a chromosome with 2 sister chromatids 1 chromosome, but then when you yank it apart, you now suddenly have 2 chromosomes of the same number. And so that is the stage where you have the duplication of full chromosomes, whereas of course the replication of the DNA material happens in S phase, where you then get the formation of the 2 chromatids, right? So let me just show you that one more time. So in S phase, chromosomes become chromatids, and then in anaphase, chromatids are separated, sister chromatids are separated, and now we have chromosomes, twice as many. All right, and that is it for today. I will see you in class.